Now we change back to quality by a presentation by Dr. Luigi Calzolai from European Commission, located in Ispra in Italy, and we hear about characterization of quality attributes of nanovaccines for COVID-19.
Thank you. So let's say the first slide. Oops. Yeah. Okay, so this is the standard disclaimer. These are my views and not those of the European Commission of the Joint Research Center where I work. So here, in this slide, I put what we know about the similarities of mRNA vaccines, LNP mRNA, and liposomal drug formulation. So obviously, they are around 100 nanometers, same size. The API, you have the doxorubicin, you have the mRNA. And if you look at they have several lipid components. Actually, apart the fine details of the chemical structure, 3 of them are the same. The only difference is in the LNP mRNA, you have the charged ionizable lipid. They have also another similarity. It's very easy to draw these very nice cartoons that probably don't represent the reality, as we have seen today. If you have seen the probably cryo-TM images of empty loaded LNP that was presented by Pfizer. So we do know that lipid therapeutics, RNA therapeutics are challenging, are complex systems. And here I'm using a classical figure from Sven Evenborger's synthesis here. Because, why? Because these systems, they have a lot of properties that can influence the biodistribution, so means the safety and ultimately the efficacy. Again, we can discuss about the fine-tuning of the different properties, but a lot of them are the same. You have the pegylation, you do have the drug loading, API, mRNA, how much is free, how much is bound, and you always have there the size distribution, or better, the particle size distribution. And so here, because of the experience we do have with the characterization of nanomedicine. So last year we published with my collaborators this proposal, the characterization strategy for nanovaccines. We published this in National Nanotechnology. And then, okay, it's, as you can imagine, it's not, you can think in terms of to build big blocks in terms of physical chemical characterization, in vitro testing before going into in vivo animal testing. In the physical chemical characterization, you have the usual suspects, the stability, especially the aggregation, the thermal stability, that we know it was a key point for the mRNA vaccines. On the chemical, you have the composition in terms of the identity of the lipids, the quantification that sometimes is not what you put in the reaction vessel or whatever, the surface charge, the amount free bound cargo. On the physical side, you have the particle size distribution. But also, we want to make this, and we wanted to point it out, you need, when you start a project, you need to have a method that is able to tell you the batch-to-batch reproducibility. So what you measure today is the same of what you produced yesterday. And this is often not a given. So I think we have a pretty good idea of what are the quality attributes of these systems. We can discuss on the fine details, but we do have an idea. But here is coming the analytical challenges. So which technique you are using for measuring a specific property or specific quality attribute? We heard several times about orthogonal measurements. Actually, this comes sometimes as a requirement from regulators. But when two measurements are orthogonal, and how to measure them, and sometimes we even use orthogonal complementary methods. Well, there is a difference, and we need to discuss and to have a good idea, a good definition of what is an orthogonal method for a given property. And then on the edge, on the cutting edge, if you want, of the field, is how to measure interface chemistry, how to measure structure. We were challenged, as I say, academic sector today by Pfizer, how you measure the structure of this complex object. the density of the PEG molecules, or these difficult organics of nanoparticles. Here we do have hope for time of flight, secondary mass spectrometry, X-ray, photoemission spectroscopy, and then I have to add also SAX, that it was not on my radar, I have to say. But most important is we are missing reference material and documentary standards. So here is our contribution to the field, if you want, developing fluffy flow fractionation to measure the particle size distribution. I'm using this example, was developed this for doxy, liposomal drug formulation, because it gives a very good, nice example of the process. We started from very well done SOPs developed inside the EUNCL project. We went on with a peer review publication together with the EUNCL colleagues and colleagues from the National Institute for Standard Technology. And now we continue working inside the ASTM standardization organization, inside the E56 group, and soon, hopefully, we have an official test method for analysis of liposomal drug formulation using multi-detector fluffy flow fractionation. We didn't stand still. We do know that we can use also for measuring LNP RNA formulation with a little bit adjustment in the separation channel. And this is unpublished data, again, with our collaborators at Syntef and PostNova. Here, we had 2 different formulations. And especially for the second, we had 3 different batches. And without even knowing what it tells you, those complex pictures, it's clear immediately that the sample 3, bottom right, is different from the other. And we couldn't detect those differences with some of the orthogonal sizing techniques we use with these samples. This is another one that I find actually essential, where there should be a little bit more of development, and the in vitro potency of LNP mRNA. It's a good surrogate endpoint of the quality, overall quality of the formulation without the use of animal testing. And, well, I need to skip a little bit on this. So the coming challenges. We heard in these 2 days already, more complex systems. Especially active targeting, attaching a biomolecule on the external surface of the lipid nanoparticle binding to a specific cell or tissues. And here, this is a cartoon picture, so it's clearly an IgG monoclonal antibodies. But also we heard a lot of more complex RNA cargo for gene editing, and so encoding the Cas9 nuclease plus the guide RNA, self-amplifying RNA. So the outlook. We have seen that the very fast approval of the mRNA vaccine, and it sort of opened the gates for the field. And we see a lot of application in different therapeutic areas. We have cardiovascular, oncology. More complex system that will come, active targeting, gene editing. So we do need the analytical method to accurately measure the quality attributes of these systems. But we also need the reference material, the documentary standards. They are needed, they are essential to give you confidence in the measurements. And we heard from Gerrit Borchardt, is the chair of the mRNA vaccine group or European Pharmacopoeia, where we are developing part of this documentary standard. And then I leave you with a question, rather. A point is how to move faster for here. Because I think there is a chance to move faster as a community to develop this. And finally, thank you for your attention. This is the Joint Research Center in Ispra in northern Italy on the Lake Maggiore, not far from here. Thank you.
Okay, thank you very much for this presentation. I think we cannot underline enough the importance of characterization and quality attributes, so thank you very much for that.